So explain the structure of mitochondria. It is one of the important question from this chapter. So first, unless or until here specifically stained this mitochondria, so we cannot observe under the microscope. So staining with the Janus green stain, so we can clearly observe the structure of mitochondria under the microscope. Here, the number of mitochondria per cell is variable depending on the physiological activity of the cells. That means the number of mitochondria varies from cell to cell. And here the typically it is sausage shape or cylindrical shape. Sausage shape or cylindrical shape. Sausage shape or cylindrical shape. So having a diameter of 0.2 to 1 micrometer and 1 to 1 micrometer in length. So remember the numericals for objective point of view. 0.2 to 1 micrometer in diameter and 1 to 4 micrometer in length. So this is the structure of mitochondria. This is the structure of mitochondria. So it is a double membrane bound organelle. It is a double membrane bound organelle. So here with the outer membrane and the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment, outer compartment and inner compartment. So this is the outer membrane and this one is the inner membrane. So this is the outer membrane and inner membrane. So this inner membrane have so many infoldings. So these infoldings are termed as the crista. Okay. So inner membrane. So along with the inner membrane, so there is a intermembrane space is also present. Intermembrane space. Intermembrane space. So the inner compartment is filled with the dense homogeneous substance called the matrix. Called the matrix. Called the matrix. So this is the matrix. And here the outer membrane forms the continuous limiting boundary of the organelle. Continuous limiting boundary of the organelle. The inner membrane forms a number of infoldings called crista. So number of infoldings are present in this mitochondria called as crista. Right. So the two membranes have their own specific enzymes associated with the mitochondrial function. So about this specific enzymes we are going to talk in plant physiology. Right. So mitochondria are the sites of aerobic respiration, very very important. They are the sites of aerobic respiration. Aerobic respiration, that is they produce cellular energy in the form of ATP. So here Krebs cycle takes place in the mitochondria, that is TCA cycle. So hence they are called powerhouses of the cell. సో ఇది మీరు చిన్నప్పటి నుంచి చదువుకుంటూ వస్తున్నారు లోవర్ క్లాస్ నుంచి. పవర్ హౌస్ ఆఫ్ ద సెల్ అనేది మీకు మైటోకాండ్రియా అనేది మైండ్లోకి వస్తుంది. రైట్. సో ద మ్యాట్రిక్స్ ఆల్సో పొసెస్ సింగిల్ సర్క్యులర్ DNA. The matrix also possess single circular DNA. Single circular DNA, very important. Whereas chloroplast contains double circular DNA and it also possesses ribosomes. These ribosomes are 70S ribosomes, whereas in eukaryotes we will find 80S ribosomes. But in mitochondria and chloroplast we will find 70S ribosomes, which are equivalent to the ribosomes of prokaryotic cell. So regarding the mitochondria, so draw the diagram and label it neatly and write some important points. That is outer membrane, it is a double membrane bound, cylindrical shape, sausage shape, and then numericals, right? 0.2 to 1 micrometer and 1 to 4 micrometer in length. And here outer membrane, inner membrane, and the inner membrane possess some infoldings called as crista, and homogeneous cells are present, homogeneous substances present, that is called as a matrix, right? And they possess some enzymes which are associated with the respiration. So they are the sites of aerobic respiration, and cellular energy is produced in the form of ATP, right? Hence they are called as the powerhouses of the cell. So they possess single, it is a previous NEET question, they possess single circular DNA, and ribosomes are present. They are 70s ribosomes. Okay. So and mitochondria divides by means of fission. Mitochondria divides by means of fission. Right. Next, another important question: plastids, that is chloroplast. So chloroplast. So chloroplast is a double membrane cell organelle. So chloroplast is double membrane. So this is the double membrane cell organelle. So here that fluid matrix, fluid substance is known as the stroma, stroma, and these are the grana of thylakoids. So these grana of thylakoids are connected by stroma lamellae, stroma lamellae. So here, so this is the stroma. So where dark reactions takes place. So regarding the chloroplast, we are going to discuss so many points in the plant physiology, right? So this one is a outer membrane, and this one is a inner membrane, and these are the thylakoids. So grana of thylakoids, and this one is a stroma, stroma lamellae, stroma lamellae. And here the number of, just like mitochondria, the number of chloroplasts varies from cell to cell. So if you take some algae, they possess only one chloroplast, and in the mesophyll cells of plants contains 20 to 40 chloroplasts per cell. Right here, the shape of the chloroplast, discoid, ribbon, oval shape. So the shape also varies from cell to cell. Next, size. So here, so 5 to 10 micrometer in length and 2 to 4 micrometer in width. And like mitochondria, the chloroplasts are also double membrane bound organelle. So of the two, the inner chloroplast membrane is relatively less permeable. It is very important for the objective point of view. Of the two, the inner chloroplast membrane is relatively less permeable. Very, very important for objective point of view. Inner chloroplast membrane is less permeable, less permeable. The space limited by the inner membrane space chloroplast is called as stroma. So this region is called as stroma. So where the dark reactions takes place. So about the light reactions and dark reactions, we are going to discuss in the plant physiology, right? So here a number of organized flattened membranous sacs called thylakoids. So these are the thylakoids. These are the thylakoids, and here they thylakoids are arranged in stacks like the piles of coins called grana, or the intergranal thylakoids. They are also called as intergranal thylakoids, and they are linked by a tubule-like structure called as stroma lamellae. This is the tube-like structure called as stroma lamellae. And here, in addition, there are flat membranous tubules called stroma lamellae connecting the thylakoids of the different grana, right? So here the membrane of the thylakoids enclose a space called lumen. So lumen. So this part is known as the lumen. So where the water splitting takes place, that is in photosynthesis, we are going to discuss about in detail, right? And the stroma of chloroplast contains enzymes required for the synthesis of carbohydrates and proteins. That is in the here dark reaction takes place. The synthesis of sugar and proteins takes place in the stroma region, where so trapping of sunlight occur in the thylakoid, right? So it also contains small double stranded, very important, small double stranded circular DNA, whereas in mitochondria it is single circular DNA, and ribosomes are present. These ribosomes are 70s ribosomes, 70s ribosomes. So the ribosomes of the chloroplast are smaller than the cytoplasmic ribosomes. That is, in eukaryote, cytoplasmic ribosomes are 80s ribosomes, right? So next, another important question from this chapter is that is yeah, types of chromosomes based on the position of centromere. Very, very important this question. So types of chromosomes based on the position of centromere. So here, so this is a primary constriction, centromere, also called centromere. So primary constriction, and this centromere is covered by a disc-like structure called as kinetochore. Kineto, kinetochore, kinetochore, right? So based on the position of centromere, we have different types of chromosomes. So that is metacentric. So here, this diagram indicates the metacentric chromosome, metacentric chromosome, and this indicates sub metacentric chromosome, and this indicates acrocentric chromosome, and this one indicates telocentric chromosome, right? The metacentric chromosome has middle centromere forming two equal arms. So these are the two equal arms, two equal arms, two equal arms. This is the middle centromere. It is a primary constriction, right? And the submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into one shorter and one longer, one shorter, one longer arm, one longer arm. So these arms are P arms and Q arms. And here another one is the acrocentric. In the acrocentric, we will find one arm is extremely short, that means extremely very short, extremely very short, and another arm is extremely very long, that is acrocentric. And telocentric is it has a terminal centromere, that means one arm is absent, that is, this is a telocentric chromosome, and some chromosomes possess secondary constrictions, and these are nothing but they are called as satellite chromosomes. So based on the position of centromere, we have four types of chromosomes. Very, very important. This question repeatedly asked many times in the examination, IP examination. So remember the size of the arms based on the position of the arms, that is mainly based on the position of centromere, P arms and Q arms. So in some metacentric chromosomes, the arms, equal arms are present, whereas in submetacentric, one is short, one is long, whereas in acrocentric, one is very extremely short, one is extremely long, and in telocentric, one arm is completely absent. Right. So this is about the cell, the unit of life. So ఈ questions perfect నేర్చుకోండి. These are the important questions from this chapter.